Good evening. Today, I, Dr. Anila Sharma, I'm thankful to the SABS team to give me this opportunity to present three very good posters, which were initially presented by Dr. Rinat Zilson from Dana-Farber, and she's a medical oncologist and a clinician scientist. These are the three abstracts which will be covered. So starting with the first poster, the ER receptor is a key driver and a therapeutic target in ER-positive breast cancers. And ER transcription is tightly regulated at various levels, be it ligand level or crosstalk between the receptor tyrosine kinases. ESR1 alterations are activating missense mutations are the Y537S and D538G, which lead to constitutive ligand independent activity, and they confer resistance to estrogen deprivation and relative resistance to tamoxifen and fulvestrant, leading to a metastatic setting. The crosstalk between ER and the other nuclear receptors or transcription factors regulate ER and transcription. This can be in the form of decrease in the ER activity by competitive DNA binding, squelching or transcriptionary machinery, or increase in ER activity by cooperative DNA binding, tethering, or chromatin opering by the pioneer transcription factor. So FOXA1 is a pioneer transcription factor that facilitates ER binding, and FOXA1, it opens the condensed chromatin by displacing the H1 histone, and it dictates increased ER binding. FOXA1 alterations can either be in the form of mutations or amplifications. This is the first poster which was presented by Dr. Liu from Dr. Schiff's team. And the topic was the targeting the FRA1-dependent transcriptional nexus in a high FOXA1-driven endocrine-resistant and metastatic breast cancer. They first studied the FOSL1, which is encoded by FRA1 gene by chip sequencing and RNA sequencing, and is FOSL1 is the top-ranked component of the AP1 complex induced by FOXA1 engaged super enhancers. Further, seeing at the Western blots at the protein level, it shows that genes are upregulated in the tamoxifen-resistant cells, and SOX9 and KLF4 are downregulated, whereas there is silencing of the FRA1. So integrating the systromic data, it identifies the downstream transcriptional targets of high FOXA1 AP1 signaling. And there was an overlap between the secretory proteins, secretome 25, and genes FOXA1, FRA1, and CJUN, which were downregulated. The secretome 25 was significantly upregulated in ER-positive metastatic versus ER-positive primary breast tumors. The silencing of the FRA1, it reduces pro-metastatic phenotypes by diminishing the clonogenicity, the soft agar colonies, and migration. So to conclude, FRA1 potentially forms a feed-forward transcriptional access to amplify the high FOXA1-induced enhancer programming that activates a pro-metastatic secretome, making it a potential therapeutic target. Now, coming to the second poster, which was inclusive of the androgen receptor. So 90% of the ER-positive breast cancers have AR receptors, and AR activation in presence of estrogen leads to reprogramming and repression of the ER-regulative proliferative genes through squelching of the co-activators and leading to tumor suppression. And as was seen in the initial paper in 2021, that AR expression was shown to be associated with good outcomes in ER-positive breast cancers. However, the older papers, they showed that AR overexpression is associated with endocrine resistance, and AR inhibition reduced estradiol-mediated proliferation. Now, these contradictory role of AR may be due to the contextual ER functioning, and was the basis for the second poster, which is mutant ESR1 receptor antagonize the tumor suppressor function of the androgens. This was the aim of the study, and through this study, the authors did the transcriptomic in ER mutant and wild type cells, hormone deprived, estrogen stimulated or androgen stimulated setting, and as expected, the gene set enrichment showed that the mutant cells showed increased signaling in the hormone deprived setting. Secondly, there was significant redistribution of AR or ER-AR binding sites in the ESR mutant cells, as was seen in CRISPR. The genes associated with overlap between ER mutant, ER-AR co-binding peaks, they showed differential expression in the mutant cells, and 65 genes showed worse outcome in the primary ER-positive breast cancers. Several genes from the 65 gene signature have increased expression in metastasis versus primary breast cancers, and ER binding density of the mutant ER-AR share binding sites in the estrogen-stimulated conditions were highest in the patient tumor samples of metastasis. So this was the conclusion, and they finally concluded that ER mutations decrease AR signaling in the estrogen conditions, and loss of AR tumor suppressor function may help metastasis in ER mutant tumors through collaboration between AR and mutant ER. Now coming to the last poster, which is ESR1 mutant breast cancer show elevated basal cytokeratins and immune activation. There was a PCA plot based on PAM50 gene expression and wild type and mutant models in 95 breast cancer cell lines, and they showed that in the PCA plot that the mutant cells, they clustered with the basal cell lines, and further there was increase in the basal genes compared to the luminal genes in the mutant cell models as well as in the metastatic breast cancer cells. The basal cytokeratins were the leading enriched basal markers as seen on the fold chain correlation of the basal genes and on qualitative PCR. And basal cytokeratins are expressed in the cells with low ER expression. The expression of the basal cytokeratin is not dependent on ER binding and is increased with ER downregulation, as seen by the genomic chip sequencing at chromosome 12, which harbors the basal cytokeratins, and there were no ER peaks seen in the wild type or the mutant cells. See, looking at the first picture, the basal cytokeratins are located within a genomic locus of chromosome 17 that has CTCF and cohesion binding in the first portion of the picture, this in the middle portion, and the super enhancer and PR binding as can be seen in the lower third of the picture. There is increased binding at the CTCF peaks in the mutant cells compared to the wild type cells, and the basal cytokeratin expression is dependent on PR. The basal cytokeratins, high ER positive breast cancers and ER mutant breast cancers are commonly enriched immune activation pathway as shown in the fold chain correlation of immune genes, and the S100, A8 and A9 were the top immune related genes enriched in the basal cytokeratin high ER positive breast cancers and ER mutant breast cancers. So summarizing everything together, they propose this working model where presence of ER mutation decreased estrogen receptors and PR at the super enhancer site upregulates the basal cytokeratin CTCF chromatin loop, which leads to the expression of the cytokines. To conclude, the ESR mutant cells and tumor show enrichment of basal subtype markers, which is led by the basal cytokeratins, and gain of the basal cytokeratin associates with the activation in ESR mutant cells via S100, A8, A9, TLR4 paracrine signaling crosstalk between epithelial and stromal cells. Thank you.